Hi, I'm Madeline Labott, a second-year PhD student in the Venutza Lab within the Center for Regenerative Medicine at Boston University, and today I will be presenting my research where I have focused on the spatial profiling of the hematopoietic stem cell niche in the human fetal liver with the purpose of unraveling the extrinsic signals controlling engraftment potential. So to understand why we are researching this topic, I will first introduce hematopoietic stem cell transplants, or HSCTs, which are a powerful therapeutic that offer a cure to a variety of debilitating blood and immune disorders. These treatments rely on the transplant and subsequent engraftment of donor hematopoietic stem cells, or HSCs, into a host. The success of these transplants is directly related to the ability of the donor HSCs to successfully engraft. Additionally, the number of patients expected to receive this procedure is expected to rise with the advent of FDA-approved gene therapies, a process in which patients are treated with their own genetically modified HSCs, following isolation, gene editing, and reintroduction of the edited cells. Unfortunately, the ex vivo phase that is associated with this process has been proven to decrease the engraftment potential of HSCs, therefore limiting the success and safety of these procedures. It is therefore imperative to decipher the mechanisms underlying engraftment and stem cell functionality to better design ex vivo culture systems that support the unique capabilities of these cells. In attempts of improving ex vivo culture systems, we look to the fetal liver, as it represents a unique developmental time point where newly emerged HSCs travel to acquire maturational cues that allow them to contribute to long-term multilineage hematopoiesis. Importantly, fetal liver HSCs have been described as having superior engraftment potential in comparison to other HSC sources at other developmental time points. Our lab has previously profiled and characterized a population of highly engraftable HSCs within the fetal liver, which allows us to obtain their unique intrinsic molecular signature, which provides us with novel insight into the pathways and factors that are enriched in the most functional subset of HSCs. Additionally, from this, we were able to identify endothelial protein C receptor, or EPCR, as a functional marker for engraftment, allowing us to recognize highly engraftable HSCs within their niche environments. However, it is still not understood what the niche-specific factors are that are supportive to these highly engraftable HSCs, and subsequently how this environment functions to promote the pathways and genes that we have proven to be important within highly engraftable HSCs. To address this, we have utilized immunofluorescence staining and spatial transcriptomic analysis. So first, we sectioned fetal liver tissues at a width of 10 micrometers, and this was used either for IF staining, where we detected for EPCR, which is our functional marker for engraftment, CD45, a pan-hematopoietic marker, and CD133, a hematopoietic stem and progenitor cell marker. Candidates for highly engraftable HSCs would co-express all three of these markers. We utilized two different spatial transcriptomic platforms, also using our fetal liver tissue sections at a width of 10 micrometers. One of these was Spatial Genomics, which is a probe-based sequencing platform, which allows us to gain insight into the specific locations of certain transcripts within the context of our tissue sections. And the last one was Mirexi's stereo sequencing, which is a platform that offers transcriptome-wide information into the location and identity of transcripts within a tissue section. So first, we had to optimize a protocol for staining human fetal liver tissue because this is an extremely rare tissue type. So we had two different types of tissues, fixed frozen and fresh frozen, and we found that we could best visualize our proteins of interest when using fresh frozen tissue that was fixed in methanol in comparison to fresh frozen tissue that was fixed in PFA. And when first optimizing this protocol, we just detected for EPCR and CD45. And moving forward, we used fresh frozen tissue fixed in methanol for all of our IF staining. So once we developed this protocol, we moved on to looking within our tissue sections to identify candidates for highly engraftable HSCs. And we found that there were several cells within our tissue sections that co-expressed both CD45 and EPCR, giving us the knowledge that we are able to identify these rare EPCR-positive hematopoietic cells within the context of our own human fetal liver tissue sections. And we can see a broader image up here and then more zoomed in on the bottom. Interestingly, we also noticed that these EPCR positive hematopoietic cells were appearing in close proximity to endothelial cells, which can be identified via their expression of EPCR but not CD45. And we noted that they appear near these large vessel regions, and we can see one of these examples closer up here, both expressing CD45 and EPCR. Additionally, we noted that some of these EPCR positive hematopoietic cells were also appearing closer to these endothelial cells that matched a morphology of a liver sinusoidal endothelial cell. And this was exciting to see because this matched previously published research which has stated the importance of both large vessel regions and sinusoidal endothelial cells in supporting hematopoietic stem cells within the fetal liver. So to further strengthen identification of these candidate HSCs, we added CD133, which is a hematopoietic progenitor marker, to strengthen the identification of HSCs with high engraftment potential within the fetal liver. And we noted two examples just within this image where we could see co-expression of all three of these markers. One of these being here, and we can see the expression of both CD45, EPCR, and CD133. Here's the second example where we can see the expression of all three of these markers again. Additionally, after we noted this, we wanted to further explore the microenvironment surrounding these cells, so we moved on to spatial transcriptomic profiling. So the first spatial transcriptomic profiling that we utilized was spatial genomics, and this is a probe-based sequencing method, which means that we were able to detect for the expression of transcripts that were included in this panel of probes, and we were able to get their exact location and also their expression and identity. And based off of this, we were able to define cell types that were present within our fetal liver tissue sections. And we noted the presence of important niche cell types within these tissue sections. We also noted that there was some interesting morphology specifically pertaining to the endothelial cells, suggesting that at this developmental time point, we're starting to see the development of these vessels, which was in line with what we were visualizing within our IF data as well. However, with this probe-based sequencing method, we were not able to gain as much insight into the highly engraftable HSCs because the transcripts that were needed were not included within this probe section. So to address this, we used a second spatial transcriptomic sequencing method, and this was Mirexi StereoSeq, which allowed us to gain transcriptome-wide information into our fetal liver tissue sections. And this method relies on the identification of poly-A tails that are present on mRNA transcripts. And then from there, these mRNA transcripts are sequenced, and also their location is noted, so you're able to detect both their identity and their location. And in our preliminary analysis of this, we were able to detect good sequence events throughout the entire tissue section. Additionally, we were able to note the expression of potential HSCs in close proximity to these large vessel regions, and we were able to identify potential HSCs by looking for the co-expression of canonical hematopoietic stem and progenitor cells. And we can see in this image the cells that are more red or orange are these candidates for hematopoietic stem cells. And we can see that they're appearing in proximity to what appears to be a vessel region, which was in line with our immunofluorescence data. So from here, we were able to conclude that our IF staining revealed the identification of candidate HSCs within the context of human fetal liver tissue. We were also able to note that these HSCs appeared near endothelial cells, both these larger vessels and also some cells that matched the morphology of a sinusoidal liver endothelial cell. We were able to validate our IF results using spatial transcriptomic efforts, and we were also able to identify relevant niche cell types using our spatial transcriptomic efforts. So within the future, we're hoping to build upon this even more and strengthen the identification of candidate HSCs by combining immunofluorescent staining and a spatial transcriptomic platform known as Xenium Spatial Transcriptomics, which would allow us to perform both IF and spatial transcriptomics on the same section of fetal liver tissue to get a really good understanding of the niche environment supportive to these highly engraftable HSCs. With that, thank you for listening to my presentation.